Alright, in this video we're going to learn to use the Codon table. Now, this table I will give to you on the test, or you're going to be doing it at home, so you can just download it. But it's going to be online for you, and you can just search in Google Images if you want a copy of it, and just go Codon table or genetic code, and usually an image very similar to this one will come up. Now, how do we use it? Well, what it does is it tells you the code used by the ribosome to read the mRNA. So let's make up a fake mRNA here. I'm just going to type some letters out. A, G, C, T, A, G, C, T, A, G, C, T, A, G, C, T. Same thing over and over again. And let's say that's our mRNA. That is the, actually it wouldn't be mRNA, would it? This has T's in it, so this would be DNA. For it to be an mRNA, we need to do what? We need to replace the T's with U's. So I'll go through here and replace these T's with U's, and that will make it an mRNA. Oops, undo that. All right, where were we? Here we go. All right, well, we have an mRNA now, and let's say this goes, and what will happen, as you've seen in the other videos, the ribosome will attach over here on the end. We usually draw it on the left, and it will move to the right, and it will read this. All right, and it would go down through here, and we saw in the video that you normally think of it as reading these three at a time. So you would say, well, it's going to read this one, and then it's going to read these three. These are called codons. When you read these three at a time, this would be called a codon. That would be one codon, then it would jump three ahead and read the next three. That would be the next codon. Well, the thing is, it actually doesn't work quite like that. There are a few more rules. According to those rules, though, if we ignore those rules, how would this be read, though? It would be GCU. So what amino acid would be placed as the first amino acid if this were actually read by or translated, I should say, by the ribosome? Well, you would go over here and look for G, C, and U. So let's see here. This is all U's, the first letter here. Notice they're all U's. This one is all C's, A's, and G's. So this is a G, so we're going to go down here, G, and then here's CU. So this is ALA, that's alanine, all right? All you'll have to do is type the three-letter code in. So we would say, okay, the first amino acid in this one would be alanine, ALA, all right? And then the second one added, let's use dashes. I like dashes better. All right, what's the next three? Well, that's AGC. So we go over here and AGC, and that's SER, serine. So you would type SER. And then you would go, okay, that's the first three, the second three, the third three is UAG. Where is it? UAG. And it says stop. Well, stop means stop. Stop making it. And that would mean that our protein would just be alanine and serine. So this is the first thing to learn, is that there's something called a stop codon. And the stop codon means stop copying. Stop the recipe. Stop mixing. If you think in terms of Grandma's cookbook, you're adding ingredients, and anytime you come to a stop codon, you say, stop adding ingredients, it's time to cook. It's time to fold the protein. So in this particular case, your protein would only consist of two amino acids, alanine and serine. You're not going to have to know those. You just have to know to write down the letters here, ALA and SER. And that would be it. That's the end of your protein. So we've learned our first lesson is that there's something called a stop codon. All right, that means if you come across three letters that are what? We came across AUG. What was it we came across? UAG. So UAG, that is a stop. There are 3 stops, all right, that we need to learn. They are UAG, all right, UGA, all right, and what's the other one? UAA. Anytime you come into the ribosome tries to read any of these 3, then it will stop reading. It will come apart, everything falls apart, and you have finished making your protein. Well, I said there were extra rules. This is one of the extra rules, but there's also something called a start codon, which is actually a little simpler. And that codon, if you look over here, it is AUG. So let's find the A's. AUG. Now here it says it's methionine, and it is MET. All right. It codes for MET, but this is always the first amino acid in your protein. It will not start reading, all right, or translating until it comes to MET. So you could think of it as a capital letter. If you were reading a paragraph or a sentence, you don't read until you come to a capital letter. You might have a bunch of nonsense letters, but you don't start reading till you get to a capital letter, which means it's the start of the sentence. Well, that's kind of what methionine is. Methionine can be later in the protein as well, but it's always the first one. So you look through here and you read the first 3, and it's AGG. Well, the thing is, I can tell you right now that there is not an AUG all the way through this. Here is an AUC going backwards, but we're going this direction. So the idea here is there is no AUG, so the ribosome would attach here, and it would read and read and read, read. There are no AUGs, so therefore this is not translated. This would be nonsense. So it would be like the cook, if you have the cookbook, and you're reading the cookbook, and it's in a foreign language, you can't understand it, you don't start mixing things. So this particular mRNA that we created doesn't work. So how do we make it work? Well, we have to start with a start codon. So let's look up here and see one that's relatively easy for us. It has to be an AUG. So why don't I just start it right here and make that an AUG? That is now the start codon. So therefore, the ribosome attaches, and it doesn't do anything. All right, now it does, when it starts out, it reads one at a time. So it's going to read the first 3, AGG. But then it's going to read it GGC. Then it's going to read it GCU. So when it begins, it's reading them one at a time. And it's not going to do anything until the first time that it gets here and it reads an AUG. And when it reads that, that means that's going to be the first amino acid in our protein. And that will always be the case. All proteins begin with methionine because the ribosome doesn't start reading until it gets to an AUG. And it doesn't necessarily have to be like 3, 3, 3, 3. When it begins reading, it's just reading one at a time. But once it does read an AUG, then you start reading them 3 at a time. This is called the reading frame. So once you find this, all right, now you know that you're going to read them, these 3. Then we're going to read CUA. Oops, let me undo that. There we go. CUA is the next 3. So let's go over here and find it. CUA is leucine. CUA, LEU. So we'll type that down. LEU. All right. So CUA, the next 3, let's see, 3 should be GCU. So that would be GCU, ALA, alanine. All right, and let's see, GCU, AGC is next. So we go over here, A, then we go over here, G, and what is it, C? AGC, that's serine. All right, so that was AGC, and then we're done, right? No, AGC, let me start at the beginning again. It's AUG, all right, then it's CUA, GCU, GCU, AGC, that's the one we just did. Then it's UAG. UAG. Ah, look at that. UAG is a stop. So we say stop. So actually, what is our amino acid sequence for this protein? What does our protein look like? It's methionine, leucine, alanine, and serine. This is it. We do not read the ones after the stop. We also do not read the ones before the start. So the ones that came before the AUG, we ignore these. All right, the ones that come after this stop. Now remember, there are 3 stops. There's only 1 start. And you do read this one. There is no amino acid associated with the stops. It just means everything falls apart. Everything breaks up. The ribosome breaks into 2 pieces. The mRNA goes one way, and the protein goes the other way. So in this particular case, this is how we read the codon table. And let's kind of try another one. So let's say I go AAA, GGG, CCC, then I go AUG. That's simple. That's the start codon, right? AAA, CCC, UUU. Then we go UAA, and then I'll just put some U's. I'm just doing them 3 at a time because it's easier to read. Realize that it won't be this easy. They'll be mixed up instead of 3 at a time. So the ribosome will attach here, and it will read AAA. But that's not an AUG, so it's not going to start translating. It's not going to start putting together a protein. Then it's going to look at AAG. Nope. AGG. Nope. It's going to go all the way down until it gets to AUG, and it's going to add this methionine. So again, it'll be methionine. All right. Then it would read what? The next 3, which is AAA. AAA is lysine. All right. CCC, that is proline. All right. UUU, right? Yes, UUU. Phenylalanine, PHE. All right, and then UAA. And UAA is a stop, and that would be your amino acid sequence for this protein. Now realize this goes on for hundreds, thousands, tens of thousands of letters to make a protein. It isn't as small as this. But on the exam, I'll just give you something small like this, and you will have to translate it. So you'll figure out, okay, here's the mRNA that my DNA created. The RNA polymerase created this. It's going to go to the ribosome. Now I have to figure out what protein it makes. So after you get to the stop, the UAA, you don't read anything after that. These are not read, and anything before the AUG, these are not read. Only the region between here is read by the ribosome. This one is a methionine always, and this one won't code for anything. The last 3, they're the stop. All right, so what are the 4 amino acids in this protein? They are methionine, lysine, proline, and phenylalanine, or these are the abbreviations. And you'll use this little code over here called the codon table or the genetic code. It's called a lot of things. But you'll use this to figure out what the sequence or the order of your amino acids are in your protein as it's coded by that mRNA and ultimately originated from your gene. So this is how you read the codon table. I will do another video on how you move from mRNA all the way through to the end of the protein. But you need to understand this one first.